Dorsomorphin, also known as compound C, is a small molecule inhibitor that selectively targets the AMP-activated protein kinase AMPK signaling pathway. AMPK is a key regulator of cellular energy homeostasis, and its activation leads to the inhibition of anabolic pathways and stimulation of catabolic pathways. Dorsomorphin has been widely used as a research tool to study the role of AMPK in cellular processes and has potential therapeutic applications in various diseases, including diabetes, cancer, and cardiovascular disease. Dorsomorphin inhibits AMP by binding to the kinase domain of the enzyme and preventing its activation by upstream kinases. Specifically, it binds to the ATP-binding pocket of the kinase domain and blocks the access of ATP to the enzyme, thereby preventing its phosphorylation and activation. In addition to AMPK, dorsomorphin has also been shown to inhibit other kinases, including bone morphogenetic protein, BMP receptors, and transforming growth factor beta-activated kinase 1, TAC1. The inhibition of AMPK by dorsomorphin has a range of biological effects. For example, it can inhibit the phosphorylation of acetyl-CoA carboxylase, SEC, and prevent fatty acid synthesis. The inhibition of SEC also leads to the activation of fatty acid oxidation and autophagy. Dorsomorphin has also been shown to inhibit glucose uptake and glycogen synthesis in skeletal muscle and liver cells, indicating a potential therapeutic application in diabetes. In addition to its effects on metabolism, dorsomorphin has been shown to inhibit the activation of BMP receptors, leading to the inhibition of bone formation and the induction of adipogenesis. The inhibition of TAC1 by dorsomorphin has also been shown to inhibit the production of pro-inflammatory cytokines, suggesting a potential application in inflammatory diseases. Dorsomorphin has been widely used as a research tool to study the role of AMPK in cellular processes. For example, it has been used to investigate the role of AMPK in the regulation of autophagy, fatty acid metabolism, and glucose homeostasis. The compound has also been used to study the role of BMP receptors in bone formation and adipogenesis. Dorsomorphin has potential therapeutic applications in various diseases. For example, the inhibition of AMPK by dorsomorphin has been suggested as a potential therapeutic strategy for the treatment of diabetes and obesity. The compound has also been studied as a potential therapeutic agent for cancer, as AMPK activation has been shown to inhibit the growth of cancer cells. In addition, the inhibition of TAC1 by dorsomorphin has been suggested as a potential therapeutic strategy for the treatment of inflammatory diseases. In conclusion, dorsomorphin is a small molecule inhibitor that selectively targets the AMPK signaling pathway. The compound has been widely used as a research tool to study the role of AMPK in cellular processes and has potential therapeutic applications in various diseases.